And welcome to Medicine Grand Rounds. We have a number of visitors from other departments and schools even, and we're really glad you're here. It's my pleasure today to introduce Eric Green, who's director of the NIH National Human Genome Research Institute. Eric is a native of St. Louis. He grew up in St. Louis, and then a trend in his family is there are a number of Badgers, including two of his siblings. He came up to the University of Wisconsin, where he took a BS degree in bacteriology. He and I were at a meeting about the physician science workforce in D.C. about a year ago when we became acquainted, and I realized he was a Badger, and we started talking about his coming to visit with us and return home, and I'm so glad he's here. After his time in Madison, he went back to St. Louis to WashU, where he undertook a PhD and MD degree, and stayed on as a resident in laboratory medicine, which at that time was in both the departments of pathology and internal medicine. He did postdoctoral work in the laboratory of Maynard Olson, which really launched his career in genomics, and he joined the faculty for 2 years there at WashU when in 1994 he was recruited to the newly established intramural research program for the National Center for Human Genome Research, later renamed the National Human Genome Research Institute. And he worked start to finish in the Human Genome Project. He rose through the ranks to becoming director in 2009. And in his role, he's responsible for providing overall leadership for the Institute's full research portfolio and other initiatives. He led a strategic planning process in 2011 entitled Charting a Course for Genomic Medicine from Base Pairs to Bedside, which was published in Nature, and since that time has been launching a number of efforts to accelerate application of genomics to medical care. Aside from his work at the NIH, he's been involved in a number of leadership roles and high-profile efforts in genomics, including the NIH Big Data to Knowledge program, the NIH Genomic Data Sharing Policy, and the U.S. Precision Medicine Initiative. He's also been leading his own lab for the last 2 decades and has been at the forefront of efforts to map, sequence, and understand eukaryotic genomes. And he's identified and characterized several human disease genes, including those implicated in hereditary deafness, vascular disease, and inherited peripheral neuropathy. Now, he's very well published, over 352 publications in very high-profile journals, including the Journal of the Proceedings of the National Academy of Science, JAMA, and 3 of his last 4 papers were published in Nature. Along the way, he's earned a number of academic honors. Early and perhaps most important was Undergraduate Research Achievement Award here at the University of Wisconsin. He went on to receive election to membership in the American Society for Clinical Investigation, the Association of American Physicians, and in 2005, WashU School of Medicine provided him the Alumni Achievement Award. Now, Dr. Green has spoken widely, nationally and internationally, and we are so privileged to have him today join us as he provides grand rounds entitled From the Human Genome Project to Precision Medicine, A Journey to Advanced Human Health. Please join me in welcoming Dr. Eric Green.
Well, thank you very much for that kind introduction. I'm delighted to be here. As you heard, I have history of this university, being an undergraduate here in the late 1970s, and even walking around last night, driving in this morning, just floods of memories as I see various venues that bring back all sorts of reminders of my time here, some of which I would happily share with you, and some of which, as a senior federal employee in the Obama administration, I probably should not share with you. And speaking of being a federal employee, I can tell you I have absolutely nothing to disclose, because when you're a federal employee, you're pretty boring when it comes to disclosures. What I want to tell you about really is a journey that I've been involved in almost essentially starting the moment I graduated as an MD PhD student. The core of this was to try to gain a better understanding of our blueprint, the human genome. You know, a lot was certainly known about the blueprint that dictated how our roughly 10 trillion cells in the human body actually operated, but there was so much more that needed to be understood. You know, clearly we knew that these 10 trillion cells all had these nuclei that had structures called chromosomes, and certainly by the 1940s, 1950s, it was understood that DNA was this blueprint information molecule. The 50s brought knowledge about the double helical structure. The 1960s brought the knowledge of the genetic code, so we understood how DNA encoded information for proteins. But there was so much more rich information embedded in DNA. I could vividly remember as an undergraduate here seeing the early signs of the molecular biology revolution taking shape, leading to methods for DNA sequencing, methods for DNA cloning, and certainly when I was a graduate student in the 1980s, DNA cloning and molecular biology came to the fore. The tools for analyzing DNA throughout the 1980s got better and better and better, and what became very clear by the end of the 1980s was that the ability to actually comprehensively study all the DNA of an organism, the complete blueprint of humans, that is the genome, might be within reach. It was also recognized that this was a somewhat finite problem. The human genome is about 3 billion bases. Could we mount a major effort to read out a reference sequence of the human genome once and for all in some organized fashion? That notion gave rise to the field of genomics. And let me just remind you that the field of genomics, in fact, the word genomics is actually a relatively new term. In fact, the first use of the word genomics in the scientific literature occurred the year I graduated medical school in 1987. It was the lead editorial of a new journal called Genomics, where the editors actually defined the field and defined this term. It's just interesting to note that never during graduate school or medical school did I hear the word genomics once, because it didn't exist until it was finally put into the scientific press in 1987. And of course, the reason the field was named and the reason there was this excitement in the late 1980s was this idea of launching the Human Genome Project and having to get out of the gates as it did in 1990. The Genome Project, which I was fortunate enough to participate in, then as a resident in pathology, also doing postdoctoral research at the time from the beginning, was very unusual in scope and scale, very unusual in organization, international, big science, highly coordinated, highly top-down managed, laser-focused set of goals, the most prominent of which was reading out the 3 billion letters of the human genome sequence. By essentially all accounts, the project was successful, finishing 2 years ahead of schedule, so that by 2003 we had laid out at least a reference sequence of the human genome for the very first time. Well, in fact, a year ago or so, we celebrated the 25th anniversary of the launch of the Human Genome Project, and we now have, essentially in our rear view mirror, the first roughly quarter century of genomics. And it might be interesting to just remind you, and I will set this up for the first part of my talk, what were the major highlights that have occurred in genomics in its first quarter century. The first I already mentioned to you. We sequenced the human genome for the very first time as part of the Human Genome Project. And that was the signature achievement of the Genome Project. But that was 13 years ago, and a lot has happened in 13 years. In fact, I would contend that genomics as a discipline has spread across the biomedical research landscape and even beyond biomedical research. There are so many different fields of study that have been greatly and continue to be greatly enhanced by tools of genomics, a subset of which are indicated here. None of these things are what I'm going to talk about today, but they're all important areas in which genomics is greatly enriching. What I'm going to do, recognizing I'm at medical grand rounds, is to emphasize how in the last 13 years in particular, genomics is being used to understand human health, study human disease, and hopefully change the practice of medicine. I am going to emphasize that, besides just being medical grand rounds, is to recognize where I have now worked for the past 22 years, the National Institutes of Health, and in particular, I have been at this institute, the National Human Genome Research Institute, which, by the way, was created by the U.S. Congress to lead the U.S.'s effort in the Human Genome Project, but as you might imagine, with the project ending 13 years ago, we've expanded our mission to now really embrace the idea that our role at the National Institutes of Health is to think about how to advance human health through genomics research. 6 years ago, I was fortunate enough, or almost 7 years ago, fortunate enough to be appointed director of the Institute, and what I would say what we're doing in particular now as we expand this mission is to embrace the idea that what needs to happen is to think about ways genomics will be used to enhance the practice of medicine. And the framing that I like to use and we tend to use at our institute is this notion of genomic medicine. Now, by genomic medicine, I mean an emerging medical discipline that involves using genomic information as part of someone's clinical care, and of course the other implications of that use. You will hear other phrases that are often used, such as individualized medicine, personalized medicine, and later on I'll talk about precision medicine. I tend to narrow the way I describe it because I want to exclusively focus on genomic information as opposed to other personalized data we might have. So I will use the phrase genomic medicine. And what I will tell you is what we are trying to do in our institute and in the field of genomics relevant to human genomics in particular is to enable genomic medicine. Now, this is not going to be a simple endeavor. The starting line for this really was 13 years ago with the completion of the Human Genome Project, and the aspirational goal in completing this journey is to realize genomic medicine generally defined. But this is not going to be a sprint. This is not going to happen in one big surge. It's going to happen through a series of systematic developments, some of which we can anticipate and some of which we have to admit we just don't know yet. We also know that since it won't be a sprint, it'll be probably more like a marathon, and I think a marathon analogy is a good one, and it will involve a lot of people and a lot of disciplines, sort of many people running shoulder to shoulder to sort of traverse each of these steps along this path to realizing genomic medicine. Now, how do you do that, especially from a funding agency at NIH point of view, to try to actually see this visualization come into reality? What I will tell you is we think about this a lot in genomics, in thinking about how to make this progression from base pairs to bedside, or from helix to health, if you prefer the metaphor. And what worked really well in the genomics community during the Genome Project was to develop strategic plans and to publish them and to share them and to be willing to change them and to continually look at new opportunities and then recast your strategic vision. Since that worked well during the Genome Project, we were poised as an institute to lead and then publish a strategic vision for what needed to be done literally the day the Human Genome Project ended in 2003, exemplified by this Nature article. And then the Institute recognized about a number of years later that things were moving quite fast and furious, and that by 2011 it was time to renew a strategic vision. And in this strategic vision, which is now 5 years old, but is still pretty good, we directly embraced this idea that genomic medicine was an aspirational goal that we should all be aiming towards. I'm not going to go through the strategic plan, but I would just stress that it described, if you're interested in going back and reading it, and it's not bad considering it's 5 years old, it still holds up pretty well, a logical progression of what needs to be done, going from very basic information on the far left about how genomes are put together, progressing to understand how they work, using that knowledge to better understand disease, and then applying that information to advanced medical science and hopefully eventually demonstrate improvements in the effectiveness of healthcare. And as you will see in this logical progression, many of these steps in fact, have been achieved, and I will highlight these as we go along, as I return to sort of the highlight reel that I want to tell you about the first quarter century of genomics. So what are the other highlights? I want to tell you about 6 total. I've already told you about the first one. Well, I would say the very next highlight that certainly deserves very high billing was actually a direct consequence of that strategic plan we put out the day the Human Genome Project ended. And that related to reducing the costs for actually sequencing a genome. In fact, we have now, since the end of the Genome Project, reduced the cost for sequencing a human genome by nearly 1 million-fold. As I said, this did not happen by accident. In the strategic plan we published the day the Genome Project ended, we said a lot of things that were going to be needed, now having that first human sequence in front of us. But one of the more audacious things that we called for in this strategic plan were the need for technological leaps that seemed so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. And we went way out on a limb because we gave as an example the ability to sequence DNA at costs that are lowered by 4 to 5 orders of magnitude than the current cost, allowing a human genome to be sequenced for $1,000 or less. And we picked $1,000 thinking about a reasonably priced, high-powered diagnostic test. Now, the reason this was audacious, and we had our names on this paper, and we put it out there as a call to action, if you will, was that, here we were, the day the Genome Project ended, having sequenced that first human genome at a cost of about $1 billion, roughly, okay? And here we were saying we were going to come up with some technological leaps that would somehow knock 6 zeros off of that figure and yield what was known as the $1,000 genome. Well, the idea was we were going to have to get rid of the factories. I mean, this was one of the factories that produced that first human genome sequence. This was not sustainable, and it was certainly not cost effective. We needed new technologies. We needed nifty, incredibly new, novel, inexpensive technologies that would get us to that goal of a $1,000 genome. Our institute put out a major granting program to stimulate technical innovation in the academic world and even in the private sector. We deserve partial credit. And the other partial credit certainly was the private sector, where private money came in, a lot of venture capital money came in, entire companies were formed, entire industry has been formed. A beautiful example of both public and private consortium of effort, if you will, that has yielded remarkable advances. In fact, I would contend that the $1,000 genome, while we're not quite there, we're within striking distance, and it really represents, as was chronicled in this Nature article a couple of years ago, one of the most technological successes in the biomedical research arena, probably in the history of biomedicine. As shown on this graphical image on the bottom left in green on logarithmic scale is the precipitous decline in the cost of sequencing human genomes that has basically started about 7-8 years ago once these new technologies started to come on board. It's not just also the cost of sequencing, it's also the speed, especially thinking about this as a diagnostic test. So I mentioned earlier that that first human genome sequence cost $1 billion, but keep in mind, it involved thousands of people in multiple countries, hundreds and hundreds of machines, all working for about 6 to 8 years to generate one sequence. But today, the good news is that using even the currently available technologies, we can get it down probably within about $1,000 to $2,000. We will probably go south of $1,000 in the next year or so, and we could do it in about a day or 2 or 3. And the good news is that whatever technologies there are today, inevitably 5 years from now, they will be supplanted by newer technologies that are coming down. There is a whole series of new technologies at different developmental stages that I'm aware of. Some will materialize, some may not, but it's an extremely exciting time because whatever we have now, we know there's newer and different and perhaps better coming. Just for fun, I thought I would share with you one of the, if you're not familiar with genomic sequencing technologies, this is sort of one of the big rages going on these days. This is a device sort of that uses a technology called nanopores. I'm not going to describe the details, but it could be miniaturized to literally a device that you can see how big it is with a little lab on a chip component here where you simply put in blood or DNA, and you take this device and you plug it into the USB port of your laptop computer. And it is being refined to hopefully sequence a human genome in something like a day for perhaps a few hundred dollars. Now, it's not quite there yet, but this exact device was already used in the field in Africa during the Ebola outbreak to accurately sequence Ebola viral genomes. Actively been used in the field for Zika virus genomes. The point is it's working well for microbial genomes, much smaller, and it's being refined for use in larger, more complicated genomes like humans. And this is sort of, might be the future of genome sequencing. I will also point out this device is really impressive because it works equally well in a PC or Macintosh computer. So if they've solved that problem, you've got to believe they're going to solve any problems around sequencing genome. So in all seriousness, whether it is this device or some other device, better and better technologies are likely going to be seen, and already we're in pretty good shape even with what we have now. Now, one could imagine having this triumph of reduced cost for sequencing. It immediately empowered geneticists and genomicists to now not just be settled with that first human genome sequence, but to recognize that we want to sequence a lot of people's genomes, and we want to do this so that we can catalog our differences among all of our genomes. And we've now gone in 13 years from having sequenced one human genome to now having sequenced tens of thousands, and probably by the end of this calendar year, I might be able to change this to hundreds of thousands of human genomes that have been sequenced. And of course, the reason for that is that we want to gain a better appreciation for how we are different among all the letters in our genomes. A crude number is that we differ by about 1 out of 1,000 bases on average, which means we can go through and sequence a human's genome and catalog the roughly 3 to 5 million single letter differences between any two people. And by doing so, we then can develop catalogs that can be available to the scientific community. And in doing so, we then empower scientists to start to systematically go through to figure out which of those variants are biologically relevant and which ones are not. The other thing that's very important to keep in mind is that the vast majority of your 3 to 5 million variants in your genome are very common. Probably another person in this auditorium has that variant. So that the idea of cataloging at least the most common variants across the human population is certainly something that would be a doable project, and one that would empower scientists to then go through, study them, figure out which of the ones are the bad variants, risk-conferring variants, which might be the variants that are good variants, maybe be protective and so forth, or have some other positive attribute. Various initiatives have basically cataloged human genomic variation over the last 13 years. The most recent one was called the 1,000 Genomes Project, a big international consortium. Won't tell you the details of these initiatives except to give you a number. When the Genome Project ended, we had data in databases on the order of tens of thousands of places in the human genome that we knew differed among people. Fast forward to today, it's about 95 million places in the human genome. We know there are differences that commonly exist among people, and we know what those differences are. That has changed the face of human genetics because it's empowered us to go through and use that information to do studies at much greater scale and beginning to give insights about what are the variants that we should care about, including medical relevance. Of course, just because you have knowledge about variants, you then want to be able to say, what are the functional consequences of those variants? To understand that, you need to understand how the human genome actually works. I would contend the first 13 years of having the sequence in front of us to study and understand, we've made profound advances in understanding how the human genome actually functions. Now, the reason we needed to do that was because this is what the Genome Project delivered. It delivered the sequence. It did not deliver an annotated interpretation of the sequence. That was going to take decades. Actually, it's going to take generations to totally solve. We've come a long way in 13 years. We now know where are the roughly 1.5% of the letters of the human genome that directly code for proteins or are protein coding genes. We have a pretty good inventory of the roughly 20,000 genes in the human genome. But we've also had a stark realization that the functional complexity of the human genome does not reside in our genes, but rather reside in other parts of the genome that are non-coding in nature, but are performing important biological functions. These non-coding functional sequences are exemplified by regulatory elements that dictate when and where and how much genes get turned on, other elements that are important for chromosome structure, function, replication, segregation, and so forth. And this is a huge area that we still need to untangle, but we're making great strides along the way. And I would also say that with technology surges, we've had better ways of analyzing DNA. That's begun to really teach us a lot more about a whole other language of DNA, and that's epigenomics, or ways that our DNA gets decorated and modified or associated with proteins. And that association with the DNA very much influences genome function. And we have annotations on what parts of our genome are being subjected to epigenomic and epigenetic modifications. And so big projects like the ENCODE project, that's our big project for cataloging and all of these functional elements, making that available to the scientific community and so forth, are ongoing. I often contend that when the Genome Project ended, we knew about this much about how the human genome works. Now we probably know about this much, and ultimately, of course, we need to know about this much. We've made a lot of progress, but this is an area that will continue, I contend, for generations because there's a lot of complexity in those 3 billion letters, and we're just getting a cursory understanding of the functional sequences. We need to then also figure out the choreography and what inevitably is going to be a lot of complicated mechanistic aspects of genome function. But I think remarkable progress has been made. Well, with our better ability to sequence genomes, understand variants, and what the functional implication of those variants, it has provided new opportunities to now better understand the role that our genome plays in health and in particular in disease. And I think we've seen some pretty good advances in unraveling the genomic basis of human disease. The advances have been different depending upon what class of diseases we're talking about. All diseases, of course, have an underlying genomic cause or at least a genomic influence, but it differs depending upon whether you're talking about more rare diseases or more common diseases. The complexity also differs because of it. Let me review this. So in the case of rare diseases like sickle cell anemia, cystic fibrosis, Huntington's disease, these are diseases that are rare in the population, but they're at least relatively straightforward at a genomic level. It's pretty much a mutation in a single gene that is the major risk for getting the disease. There might be some environmental influence about severity and other genomic variants that might influence severity. But by and large, these rare diseases involve a single gene. They're monogenic, also known as Mendelian disorders because of the famous geneticist Gregory Mendel. Remarkable progress has gone on in understanding what are the mutated genes accounting for rare diseases. Again, some numbers. The day the Human Genome Project started, October 1, 1990, 61 rare diseases we knew what the mutated gene was, 61. Fast forward to today in particular, that number is 4,500. Remarkable progress. Now, there's still about 2,000 rare diseases we haven't figured out the genomic basis for. We have major programs to try to accelerate now closing that gap. But going from 61 to 4,500, big triumph, I would contend, the last 26 years. And now we have to fill it in the rest. But rare diseases actually are not what fill this hospital or clinics around the world. Rare diseases obviously are bad for families. They're important to study, and they're devastating to patients. But the reality is the major healthcare burden worldwide, as you all know, are common diseases. Hypertension, diabetes, autism, Alzheimer's, cardiovascular disease, and so forth. From a genomics point of view, rare diseases are hard. Common diseases are hard because it's not a single gene and it's not a single genomic variant. It is inevitably going to be multiple, in most cases, multiple genomic variants, sometimes in coding regions, sometimes in non-coding functional sequences, with what is almost certainly a greater influence of the environment. And untangling this, when you're looking at individual variants, very small contributions, gets to be a very challenging problem, but one that is getting more and more tractable. But still a long way to go. I'm not going to go through details. Some of you are probably familiar with some surging efforts in doing genomic association studies or genome-wide association studies, where very large collections of patients with or without common diseases are studied. You look at all their genomic variants, you compare it to control groups, and you try to find regions of the genome harboring a variant that seem to be inherited at a greater rate in those people with the disease compared to those without. The success of this approach has yielded thousands of regions of the human genome that are clearly harboring variants, contributing risk for common diseases. But now the stage is to try to go into those regions, figure out which is the variant that's conferring risk, how is it conferring risk, what are the biological properties of the pathways affected by it. And that's a big surge of effort right now, really across almost all disease areas for common disease effort, inevitably involving probably genome sequencing of tens of thousands of people with or without a disease, and we have major programs that are attempting to do just that because the cost of sequencing has gone down considerably. But I would contend, I think the next 10 years in particular, maybe even the next 5, you will see a lot of successes, we hope, in understanding what are the genomic variants that are contributing risk for more common diseases that really are important to understand. But I've now spent the first 27 minutes or so of this talk, and I've said nothing about the practice of medicine. This is all basic genomics, maybe into some translational work with disease. And I would contend 6 years ago, 7 years ago, when I became director of the Institute, I probably almost had to stop my talk about here because there really wasn't real examples of genomic medicine that were worth describing. But that's changed. And I would contend that there are now some vivid examples of genomic medicine that are in action that are now emerging. It is the tip of the iceberg, a lot more to come. But I think it is worth sharing this with you because I think it's highly illustrative of what we likely will see in the coming 5, 10, 20 years. I would contend in a fashion that simply wasn't the case 5, 6 years ago, genomic medicine is coming into focus, I think very vividly and I think very illustratively. I'm biased, so that's why I say this, but I think there's others that are agreeing with me. I would just toss out. There's this magazine, maybe some of you read, called Modern Healthcare. Earlier this year, they did a survey of their readers and said what were the top advances in the last 40 years. Coming up number 1 by this survey was the sequencing of the human genome. Felt that the readers thought that it represented the most significant breakthrough in healthcare over the past 40 years. It can't be because we've really changed healthcare. I think the survey simply reflected people being very optimistic that the trajectory we're on, that we will look back and say, by sequencing the human genome, we put us on a trajectory to actually change in the future healthcare, hopefully in a very positive way. So what are the hot areas? I want to share 5 of them with you. I think it's illustrative of what really are some of the most exciting, in some cases the most compelling areas where genomic medicine is emerging, some of which you'll be familiar with. I'll start with cancer. I've said nothing about cancer so far, but I actually know, even from dinner last night, that there's a lot of cancer genomics going on here at the UW and across the state. This is a sophisticated audience, medical grand rounds. You know that cancer is fundamentally a disease of the genome. And what the technical innovations that have allowed us to read out normal genome sequences have also allowed us to read out cancer genome sequences. This has gone on internationally across many projects with thousands of tumors, tens of thousands of tumors, and normal DNA have been sequenced in parallel, developing catalogs for all the major types of cancers, knowing what are the most common genomic changes seen in those tumors, and in particular then giving insights about the driving pathways leading to those types of cancer, and importantly, new diagnostic insights that are giving us new views about really the best way to diagnose cancer. You know, we're already seeing a change whereby, you know, cancer histopathology, which was the mainstay of cancer diagnostics for decades, it's important and will continue to be important, but augmenting this, of course, will be information about that particular patient's genome sequence of the tumor, and having the kind of cartoon-like signature you see on the bottom right tell us a lot to augment what is being gained by visualization of the histopathology by pathologists. It is absolutely going to be, it's the case for some kinds of cancer, and I think that list will just grow every single year in the coming decade. Again, morphology will clearly continue to be important, but genomic signatures are going to be a key part of the cancer diagnostic paradigm. Again, I'm biased, but all you got to do is look around, jump on the web, go to, and maybe this cancer center does the same thing. Lots of advertisement about genomic testing, lots of advertisement about DNA being a critical piece of the puzzle for major cancer treatment facilities in the country and actually even abroad. So cancer is an easy one. It's obvious. It's the first one lots of people name. I would contend the second hot area worth emphasizing is pharmacogenomics, one that probably is just starting to be seen for some types of medications. I think, again, that list will grow. I thought it was interesting. Some of you certainly will know this far better than I will know this, but Nature had an article about this last year talking about the imprecision of how we prescribe medications, indicating for these 10 medications, ones that I'm sure many of you are very familiar with, the individual in blue are the proportion of people that the medication works for. The individuals in red are the proportion of individuals the medication doesn't work. What's happened in genomics, especially with some of these big studies with knowledge of the common variants in the population, are studies that have begun to reveal what is the genomic basis for differential drug response. And with that, have give clues about genomic changes that are somehow influencing drug metabolism pathways or other pathways that influence how these medications work. And as a result, the notion of doing stratification of patients based on genomic information is no longer hypothetical. That for some medications, this is becoming standard of practice, and the FDA is recognizing this, and their list of drugs that have to carry an information label that talks about genomic information perhaps being valuable in making decisions about this medication for your patients. That list is over 100 and will continue to grow, and we are hoping to see a greater uptake of genomic testing in advance of decisions about what medication to give patients, or in some cases, what's the right dosing for that medication. So that's also here and now expected to grow. Also here and now, and really arriving on the scene pretty quickly over the last 5 years or so, is just the routine use of genome sequencing to deal with the diagnosis of rare diseases, and especially in cases where you simply don't know what is wrong with that individual. The sort of the prototypic cases that first emerged, actually one of them was over in Milwaukee in this state, published in 2011. This not known what was wrong with this boy until they sequenced the genome. When they sequenced the genome, immediately became clear what was wrong, leading to some treatment options that were beneficial. That same year, these twins from San Diego, same problem. Diagnostic workup, never could figure out what was wrong, finally figured it out by sequencing their genomes, and actually were fortunate enough to see some options for medications, and they're actually doing quite well now. This led Nature to even predict a year later that sequencing was set to alter the clinical landscape, and indeed by 2014 they admitted it was. And basically talking about when you have a rare disease, you don't know what's going on, it just makes an awful lot of sense to spend $1,000 or $2 to try to get genomic information. And the percentage of cases that you actually can figure out what the basis for that disease is growing every year as we get better and better understanding of how to do the sequencing, how to interpret the sequence, and be able to understand what the role that the variants that are encountered might play in that disease pathogenesis. So this has become, in many ways, mainstream probably here. I wouldn't be surprised if the medical geneticists are performing genome sequencing for some of their cases. It's also changed a lot of thinking around undiagnosed diseases. Shown here is the team at the NIH intramural program that launched the Undiagnosed Diseases program that basically has as a centerpiece component bringing in patients really from around the world who have gone, in some cases for decades, without a diagnosis, and using genome sequencing as a centerpiece of trying to do one last attempt to get a diagnosis, and doing so in a non-trivial fraction of the time. This program was so successful at NIH, we just recently nationalized it. Now there's about 6 or 7 centers around the country that are part of the Undiagnosed Diseases Network. And meanwhile, other medical centers are setting up such clinics, and I'll also tell you other countries are mirroring similar programs. The fourth hot area of genomic medicine I always like to emphasize is related to the genomics of pregnancy. There's sort of 2 stories underlying this, one at a prenatal stage, one at a postnatal stage. The prenatal stage is probably the most vivid example of genomic medicine in action. Let me just remind you that for decades, it was very routine to do prenatal genetic testing, but it involved invasive procedures, such as shown on the far right, like amniocentesis or chorionic villus sampling. My wife had that done for both of our children. It yielded, in the old days, karyotypes. In the newer days, other methods were used once you had fetal DNA to try to look at aneuploidy and try to make gross assessments about the genomic health, if you will, of the unborn child. And this was done routinely, but of course the invasive aspect of it was both unpleasant and also risked the viability of the pregnancy. This is all changing now, because these new methods for sequencing DNA are so exquisitely sensitive that they allow you to detect the very small amount of fetal DNA that is shed from the placenta into the maternal circulation. And now you can actually detect it and do the same diagnostic workup you used to do when you did more invasive methods to access the fetal DNA, by simply accessing the small amount of fetal DNA floating around in the maternal bloodstream, which means a simple blood draw is all that is needed. This non-invasive prenatal genetic testing is not a fringe activity. In fact, worldwide, through the 3 years from 2012 to 2014, well over 1 million women had this non-invasive test done. The private sector has really jumped into this, and I'm told that in 2015 the number surged over 1 million. So over 1 million women, pregnant women worldwide, are getting a genomic test done that only came about because of these new technologies of DNA sequencing. Clearly, this is genomic medicine in action, and one that I think has very much changed the practice of prenatal testing. That's prenatal. But then there's postnatal, and here the situation is probably a little bit different. Time magazine predicted a couple years ago that maybe by 2025 everyone will get their DNA mapped. I think they meant sequenced at birth. And I don't know if I agree with that, and in fact I'm not sure that's desirable. It's probably something we need to think carefully about, and indeed we are. There's a lot of logistical things you want to consider, probably even some ethical things that are important. We've actually launched a major research network in partnership with the Child Health Institute to study this. And their early results, I think, are really illustrating some of the challenges one will have to face, but also some of the great opportunities by having such genomic information about an individual starting at the beginning of their life and maybe carrying throughout their whole life. Healthy babies, sort of one category, and that's probably the one that will require extensive study. There's another category of newborns that already we're seeing genomic medicine being implemented in a very productive way, and that relates to acutely ill newborns in neonatal intensive care units. One of our grantees has really reduced to practice a pipeline involving the rapid sequencing of a newborn genome sequence in the cases where the neonatologists simply don't know what is wrong with that child, expect the child to expire within a few days, and having a methodology to be able to, in a matter of about 24 to 36 hours, get a genome sequence generated and analyzed. And we are hearing that more and more neonatal intensive care units are picking up this approach because they recognize that in some cases it is highly valuable as a diagnostic tool to give some clue on how to save that baby's life. The last hot area I want to mention is hot not because it is solved, but it's hot because it's really important and something that I bet all of you probably are going to be thinking about and are going to need to be helping guide us to make this correct. The real problem I think we're going to face over the next decade is that generating a genome sequence on a patient, it actually is pretty trivial. Generating that data is trivial. We can do it on any patient. The reality is that there's a huge gap between our ability to get the set of variants in a patient, 3 to 5 million variants, and understand what even a small fraction of those variants mean. If you generated a genome sequence on one of your patients and you rounded on that patient the next morning, this is what you'd feel like. And understandably, that's what you'd feel like. Because we have this massive gap that we need to close. Now, every week, more and more papers are published in the primary scientific literature telling us what the variants might be doing biologically. But you don't have time in your busy practice to look up those papers and to sit there and go through and try to guess whether or not they're accurate and what it means for clinical practice. We need to develop a better information system that is curating all that information. We need all that information sort of streaming into electronic health records, both the data on the patients, but also the practice guidelines and other recommendations based on curation of the tsunami of literature that's taking place. And we want to empower all of you to have a quick lookup when you encounter a variant with some little device that will tell you what the clinical significance of that variant is and what you might want to do about it in terms of your practice. We are so far from having this become a reality. In fact, we don't even know how to get there. So we started a research network to try to explore exactly that, something called ClinGen, or Clinical Genome Research, which is just, and is, by the way, it's another international effort because other countries are starting the same thing, to figure out how to develop the knowledge base to close that gap. Because if we don't do that, this will not be implemented in clinical practice, knowing how you really have to have something that is quick and very clear. Otherwise, we're going to be drowning in variant information in the primary literature. So this is one of the grand challenges we are trying to solve, and we recognize this is very important to make genomic medicine a reality. So I have now, in about 40 minutes or so, romped you through about a quarter century of genomics. The other thing that's happened in those 40 years is that the relevance of genomics has changed. When I got involved in this as a resident and a postdoc, it was just about researchers, just geeks like me working away trying to figure out how the human genome was put together and eventually sequencing it. And during the Genome Project, that's what it was. But when the Genome Project ended, I think we were successful at getting healthcare professionals to come into the tent to help us think about how this information might be useful for the practice of medicine. And I think, as I've illustrated, we're starting to see some successes. But with the successes come an additional responsibility, because when you start to use words of genomics as part of cancer care, as part of medication choices, dealing with prenatal testing, dealing with rare disease diagnostics, all of a sudden genomics becomes relevant to patients. And once it's relevant to patients, it's relevant to friends and relatives of patients, which means it's relevant to everybody. And so this is also now something that the genomics community has to help with, and we are thinking about lots of things of how to sort of help society deal with this onslaught of advances in genomics, and thinking about how to really capitalize on all these opportunities as we think about how medicine might be practiced in the future. So speaking of the future, in the last 5 or 10 minutes, I just want to briefly tell you about one last initiative that in many ways will bring everything full circle. Among the many people in society who has been monitoring advances in genomics, and it is the political season, so it's relevant to point this out, is this guy, actually, it turns out. This guy, who I hope you know is the president, at least for now, only until January 20th, though. He's actually always been very interested in genomics. Even when he was a senator, he actually introduced the bill in the U.S. Senate called the Genomics and Personalized Medicine Act of 2006. It never got passed, but it signaled his interest in genomics. And as a result, even when he became president, he really follows biomedical research and some of the key advances, of genomics being one of them. Well, this is a scientific and a political story I'm going to tell you in 10 minutes, but it might be, I wanted you to be aware of it, and I think it's very exciting. About 2 years ago, June of 2014, he actually convened a small group in the Oval Office that included the NIH director and some other key science advisors, and basically said, I really think genomics is incredibly exciting, and I think these genomic medicine he was aware of. I've got 2 years left in my presidency. There's a lot of things I won't get through the Congress, tax reform, immigration reform, but I probably could get a scientific new initiative through Congress. And maybe we want to do run around genomics. And could we come up with something and that I could sort of get launched before the end of my presidency? By the time that first meeting had ended, he was rightfully convinced not to have such a narrow laser focus on genomics, recognizing that the reason he and many others have sort of seen genomics be sort of the cutting edge of everything is because of the technology surge that have really helped us untangle this part of the equation for most diseases. But that to thinking in a forward way, there are some very exciting opportunities with new technologies that might allow us to better understand this other important component of disease, lifestyle, environment, diet, and so forth. And that maybe have a broader framing around genomics, and I actually think that was the right decision. The president ultimately decided to use the phraseology precision medicine because of that, and recognizing that it's not just the G's, A's, T's, and C's in our genome, but other elements, what we're exposed to, our lifestyle, and what we eat and so forth, all being very important for the disease process. And so this, I think, sort of gives a broader context for sort of accounting for individual variability. Again, what I'm most aware of course, are these genomic advances I've spent most of my time talking about. But there are some, and I think some of you are probably familiar with, some pretty cool new things coming up with new technologies for measuring physiology and monitoring various aspects of people's health, mobile devices and so forth, wearables, but also better ways of monitoring what people are doing in terms of their sleep and their exercise routines, what they're being exposed to, and then what they're doing to themselves, so whether what they're eating or what their destructive habits they may have. And so the idea was there are great opportunities to actually study all these domains. They're only going to get better in the coming years. Let's have a major research initiative to try to put this all together to see if that the variability about individuals might be helpful for tailoring medical care. And of course, at the heart of it, what the president recognized was that today so much of what we do is based on the expected results of the average patient. It's getting better, but it's still far from perfect. And is there a tomorrow where medical care could be more based on these differences? Genomic is getting clear, but also environmental lifestyle differences that might improve our ability to treat patients. And he said, what can I do to get us from today to tomorrow? And he asked a small group of people, I was included in that, to go and try to develop a plan in the summer of 2014 to bring back to him in the fall to perhaps launch a new initiative before the end of his presidency. We formulated that plan. It sort of ruins your summer when the president asks you to do something and you already have a day job, but that's okay. And by the way, various plans will come up with. We ripped them up, most of them, and then we finally came up with something we thought was good to bring back. It was brought back to him in the fall of 2014. He embraced it and said, develop it in greater detail, build a budget for it. I'll build it into my budget. He announced it in the State of the Union address in January 2015, and then 10 days later at this ceremony in the White House, he announced formally the Precision Medicine Initiative. Has several components, some regulatory reforms, a major surge into cancer genomics as the lowest hanging fruit. That's been mostly now uptaken by Biden's cancer moonshot effort. But the centerpiece of the Precision Medicine Initiative that you will increasingly hear about in the coming years is the creation of a U.S. national research cohort. The idea is to recruit and properly consent 1 million U.S. volunteers who raise their hand and say, I will participate in this longitudinal study. Probably will go on for decades. All the participants will share genomic data, lifestyle information, biological samples, all linked to their electronic health records. But then also share this data widely with lots of scientists and other individuals who want to investigate it, but obviously build in appropriate privacy protections. If you had such a resource, it would serve as a remarkable foundation of data that you could imagine doing studies to improve human health outcomes, fuel development of new treatment, catalyze a new era of data-driven and more precise preventative care and medical treatments. In a rapid fashion, I'll just tell you, why did all this come together? I mean, genomics is what I emphasized. But what the president realized, and I think all of us have realized, is that electronic health records, which 10 years ago were only found at 20% of healthcare delivery sites across America, now it's over 95%. That data sits there most of the time unanalyzed. It is a remarkable resource for potentially doing research if it's properly harnessed and integrated with other data, like genomic data, and then also augmented with newer data that we will be able to collect with new technologies for monitoring physiology and monitoring lifestyle and diet and environmental exposures and so forth. All of us or many people wear Fitbits as recreational devices, but I think all of you are aware those technologies are going to get better and better. Some of them are going to become clinical grade and will become part of mainstream medicine, and if we could collect lots of data on individuals, probably including collecting data that will be streaming through people's smartphones into data resources. The people recruited to this will be harnessed with such devices, and we will collect a lot of physiology data from them over many years. Be a lot of data, right? Genomic data, electronic health record data, new technologies, but that's what biomedical research is going to be about in the future. A lot of high-powered data analysis, and we need to get good at this, and we will, and this will help drive that forward. And finally, the recognition that we want to have new models, and you'll hear a lot about this as this project sort of unfolds in the coming months, where participants in this are going to be partners, they're going to be heavily engaged from the beginning, and they're going to have a lot of control over what happens with the data. Even their own individual data, they get access to it, and they also get to dictate what studies might or might not be done. There's going to be a tremendous emphasis on on participant engagement. A lot of this will be done through social media and through smartphones and the like. So I'm giving you just the simple view of this. If you want to read more, and I hope you do, this is the landing page at NIH for the Precision Medicine Initiative. Buried in these various subpages are lots of white papers and PowerPoint presentations and videos of the many months that have led up to the launch of the initiative. We did get bipartisan support. We did get new money to launch this project this fiscal year. And some of you may have seen various breaking news as we have sprinted to the launch of this. July was a big announcement period where lots of new grants went out in early July to basically stand up all the elements that will allow us to begin recruitment of individuals in the coming months. The president has never been on the sideline. In fact, it's both a blessing and a curse when the president's that interested in science. He's constantly involved in wanting to know what's happening with this. And literally the day after these announcements for these grants, he wrote an op-ed piece in the Boston Globe very much embracing this project and seeing how it might end up really advancing medicine in a very broad way. So it is going to be launched before the end of his presidency. You will be hearing about that inevitably because this will hopefully get a lot of positive attention. And then off we go. It's going to be very exciting. Now, I will also, in wrapping up my talk, just say that we don't have all the answers of exactly what this is going to look like. And there's some historic irony, especially for those people like me who sort of were once upon a time involved in another big high-profile project. It does feel, and very much what it felt like 25 years ago for me. That first year when we were standing up the Human Genome Project, there were so many aspects of it that we didn't know how we were going to do it. You would have asked me, how are you going to map and sequence the human genome? I would have said, don't know, haven't figured it out. It's really exciting. We're going to do this, and we'll work it out along the way. Same exact thing is true with so many elements of this big, audacious precision medicine initiative. We have a cursory idea of how to get started, but what this actually looks like over time, we don't know precisely. No pun intended, but we also are willing to get going and be willing to modify our plans as we go. Because at the end of the day, these big projects really benefit from not getting too rigid in the goals and being very facile as new opportunities arise. At the end of the day, these are both research projects, and we just have to admit that the best way to conduct them is to be very willing to admit you don't know everything. This is actually a sign I have above my door in my office, but I really do believe it. Nice quote by Einstein that if we really knew what we were doing, it wouldn't be called research. That's what these big projects are all about, is a willingness to embrace them as research projects. We don't know exactly what we're doing, but boy, they're awfully exciting and they have great potential for advancing human health. So I will stop there, and it leaves enough time for questions, and happy to take those questions. Thank you.
Thanks so much, Eric. That was just outstanding. I'll ask you to call on the question and repeat the
Happy
question.
to do it. I will repeat them. So, questions. So maybe back there in the aisle.
Given the potential for genomic weapons, how is the field being policed?
How is the field? So the question was concern around genomic weapons and how is the field being policed? So it's a hard, very unpacking that question. It's a lot of complexities. What I would say is that, and I think there was a lot of concern after the Genome Project about genetic discrimination, as an example, and legislation was passed, the Genetic Information Non-Discrimination Act, which helped put us on a path towards preventing those concerns. Similarly, with the Precision Medicine Initiative, lots of concerns around privacy, but that's also beneficial. It gets a lot of attention of key people, both with respect to getting appropriate policies put in place and also to enhance, to make sure that appropriate privacy protections and appropriate legislations in place that people who do things they shouldn't do with some of this data will be penalized in some way. In terms of weaponizing it, you might imply that there's concerns about will genomic insights give sort of targeted abilities of individuals, to target individuals in some way. And there is some concern about that, I can tell you. I don't think the scientific basis for it is very strong, but it is clearly something we are absolutely involved in trying to do appropriate research to understand it, but also to try to keep the conversation on firm foundation scientifically. Yeah, in the back.
You identified the lack of knowledge about the functional nature of all of these genomic variants as a real challenge. And can you comment on the role of model organism research in answering this question?
So the question was, since I readily admit that there is a tremendous amount we need to learn about the role that genomic variants play in function of genomes, in particular in in coding regions, but especially in non-coding regions, and what the role model organisms would play. What I would tell you is they play an incredibly powerful, important role. I'll give you, and very much embraced, I think increasingly embraced by human and medical geneticists, recognizing that when they have these variants of unknown significance and not knowing what to do, they are turning increasingly to their model organism research colleagues to help test what those variants are doing, to help guide making diagnostic decisions. And I'll put a fine point on it. I mentioned the Undiagnosed Diseases Program, which is now the Undiagnosed Diseases Network. So a part of that we've now put in place is which grants have been given out, include model organism cores to sort of take variants coming out from those patients and test them in models such as flies and fish and so forth. Again, directly embracing the idea that they play a critical role in understanding the biological role of some of these variants, in those cases being medically relevant variants. Absolutely. Yeah, maybe here.
Thank you. It was a wonderful talk. You alluded to some of the ethics, and as we move from the monogenic disorders, where I know whether to give abacavir or not to a patient, and we go to risk, how can we avoid giving people too much knowledge of their future and eliminating hope?
Yeah, so the question, if I could paraphrase, was, rare diseases more straightforward, pharmacogenomics more straightforward. You get into common diseases, multiple variants, very complicated, a lot of risk-based information. And how do you take away their hope? Being careful not to take away their hope by having too much information given to patients. So first thing, I completely I agree with this issue that we need to be very mindful and careful on how we present this. Notice in my hot areas of genomic medicine, I did not include any sort of prognostication about common diseases based on genomic information. I personally don't think we're there yet, and I'm not sure we'll ever get there. I'm not sure what that's going to look like. Certainly some people who are more enthusiastic about the future of genome sequence might push stronger to say that we'll make all sorts of predictions about diabetes, I'm not sure. I'm not convinced of that. I'm not going to put that on my hot list quite yet. I think the most important thing we need to do is to make sure we're informing, giving a very balanced view about what is and is not known, what is and is not clinical practice, and not let too much hype about genomic information giving us all the insights medically, because I think that's an overreach. So I think I share your sentiment that we need to be careful. The only other thing I would say, and you'll see a lot of this talked about increasingly, is I think there'll be a lot more argument about whether patients have a right to know that or not, and who should make the decision for the patients. And a lot of this has come up with some of the direct-to-consumer genomic testing that's available, and some people being very concerned, and some people saying patients have a right, and people have a right to get their own information if they want it.
We have time for one quick question, one quick answer, please.
Okay, right here then, right here. Yeah, no, you.
Talk about how the regulation of laboratory developed tests may affect the rate of progress or the others.
So he wanted a quick answer, and that's about an 8-hour answer. So the question was, and it's a great one, is that, and I can't answer it quickly, the question is, how will the regulation of genomic testing and broadly, especially FDA getting much more engaged, how might that slow progress in all of this? This is, the way I would answer it is that we're doing everything we can not to have it slow progress. 6 years ago, I can tell you, we had essentially no relationship with the FDA. I mean, to be honest with you, my institute. And I will tell you, I'm meeting with them constantly. My staff is meeting with them constantly. It is one thing we can try to do to help get them in they have a job to do, but we also have a job to help them make sure they understand some of the consequences of these things. Obviously, regulatory oversight and challenges with the practice of medicine is not new to genomics. It has been around for lots of things, but it is hard. Genomics is not. It brings up issues that have never been seen before, some complexity, and so as a result, it is challenging. But there's a lot of effort by, including professional societies, as you may know, to try to get everything we can to help the FDA do their job without slowing down progress in this arena.
With that, I want to thank Dr. Green for just an outstanding grand round.